Hi, welcome to this video on enzymes. So first let's think about a definition of an enzyme. So an enzyme is a biological molecule that is able to lower the activation energy of a reaction. So collectively that means it's a biological catalyst. But what does this lowering of activation energy actually mean? Well, if we take a look at any reaction, we start with some reactants. So in this case, I've got carbon dioxide and water. And you can see down here that after the reaction has taken place, the products have formed. And the total energy in the products is actually less than the total energy in the reactants. So this is the overall energy released during the reaction has resulted in less energy in the products compared to the reactants. But in order to get from the reactants to the products, you can see that we actually needed to input some energy in the first place. Now, this is known as the activation energy. It's the energy required for these two to react together. Now, without an enzyme, you can see that we actually have to input quite a large amount of energy. So this is a large amount of activation energy in order to cause the reaction. Once the products are formed, the energy drops right back down again. But we need to make sure that we can input enough energy to cause the reaction in the first place. Now, this is without an enzyme, and you can see that with an enzyme, the energy that we actually need to input to start with is much, much lower with an enzyme. So the activation energy without an enzyme is high, and the activation energy with an enzyme is relatively low. Okay, so that's why it's known as a catalyst, and any catalyst has this function to lower the activation energy of an enzyme. So what exactly is an enzyme? Well, you might be familiar with this model here. You can see that we have an enzyme and we have a substrate. So the enzyme itself is a globular protein. So we've looked at in a previous video at globular proteins. They are long chains of amino acids, so they're formed of polypeptides, sometimes multiple polypeptides joined together. And importantly, these polypeptides have a tertiary structure. That means that they have lots of complex intramolecular bonding taking place. So these bonds are hydrogen bonds that you can see here. So this is where we've got two partial charges attracting each other. We have disulfide bridges. So these are very, very strong covalent bonds. And then we also have ionic bonds as well. So in all cases, these bonds are taking place between the R groups of different amino acids. So hydrogen bonds being relatively weak, and then the disulfide bridges and the ionic bonds being relatively strong. So within this enzyme then, we've got this folded up polypeptide chain with all these complex bonds within, holding it in a perfect shape. And this shape needs to be perfect because one region of the polypeptide, one region of the protein, known as the active site, needs to be in an exact shape, an exact configuration to allow a substrate to fit in. Now, a substrate is just another name for the reactants that we looked at previously. So in some cases, this is going to be one molecule that fits in, and the enzyme is going to cause this molecule to break down. And in other cases, this could be 2 or 3 different molecules that all fit in simultaneously, and the enzyme is going to cause them to bond together. So yes, that's about it, I think, for enzyme structure. So let's have a look at 2 hypotheses about how enzymes work. So this is the original one that you probably will recognize from GCSE. This hypothesis is known as the lock and key hypothesis. And essentially what it says is that the structure of the enzyme, the tertiary structure of the enzyme, is going to be give the enzyme a specific shape, which is going to give the active site a specific shape. And in this model, the substrate has a shape that is perfectly complementary to the shape of the enzyme. So it's important that you can use the term complementary. It means essentially that one is a mirror image of the other. It doesn't mean that the shape is the same. It means that one shape will fit perfectly into another. So that's what complementary means. So in this model then, the active site is complementary to the shape of the substrate fits in, and once it fits in, then the reaction takes place and the products are released from the enzyme. Now this is quite a nice model for enzyme action, but it doesn't actually explain how the enzyme works, because if the substrate fits in perfectly, then what is actually causing this reaction within? Because the enzyme doesn't necessarily change shape, it doesn't add anything or remove anything from the substrate. So how does it actually work? Well, this is where the next model, the induced fit model, the induced fit hypothesis comes in. Now, what the induced fit hypothesis states is that the enzyme has a specific tertiary structure and so a specific shape, but the active site is not initially complementary to the substrate. But as the substrate moves towards the active site, the enzyme changes shape. The substrate causes the enzyme to change shape. Now, it does that because those internal bonds are going to be changing ever so slightly. As the substrate interacts with the active site, some hydrogen bonds within the molecule are going to form, some are going to break. It's even possible that ionic bonds could form and break as well. So as the substrate moves closer, the active site changes shape so that it becomes complementary to the substrate. Now, this is important because now once the substrate has fit in, the enzyme is almost acting a little bit like a bow or a bowstring where it's been pulled taut. And where it's now been pulled into this new shape, it means that it's going to be placing strain on the substrate itself. So in this case, where we have a single molecule fitting into the active site, a single substrate molecule, where it now fits in, this enzyme is going to be putting strain on the bonds within the substrate. And as it puts strain on those bonds, eventually those bonds are going to break. Now, as soon as the bonds are broken within the substrate is now no longer complementary to the active site, and so these two are released, and the enzyme returns to its normal shape. Equally, if we're talking about 2 or 3 substrates fitting in and the enzyme is responsible for causing them to bond together, the same is true. As these molecules move towards the active site, the enzyme changes shape, and now it forces the 2 or 3 substrate molecules closer together. And since they're now forced close together, that means they are more likely to react together, and so a single substrate molecule will leave the active site and the enzyme returns to its natural shape. Now, it's very important you understand and are able to describe and explain the induced fit hypothesis. This one crops up quite a lot. Right, let's have a think about some limiting factors for enzyme activity. So the first one is temperature. Now, this I'm bringing up first because in order to understand temperature, you need to understand a little bit more about how enzymes work. Now, enzyme activity is all down to luck. It's all down to randomness. Now, what we say is that when a substrate collides with an enzyme, it needs to collide in the correct orientation. That is to say that when this substrate here collides with the enzyme here, this substrate needs to be facing this direction and it needs to be this way up. If it's not facing this way, if it's the wrong way up, then it's not going to be complementary to the active site. Equally, when the substrate collides with the active site, it needs to be colliding at the correct speed. If it's too fast, then it might just rebound off. And if it's too slow, then it might not cause the bonding within the enzyme to change. It might not cause the active site to become complementary. So the speed and orientation at which this collides with the enzyme is very, very important. Now, speed of molecules is dictated by the temperature of the molecule, because as you increase the temperature of the molecule, that is going to increase the amount of kinetic energy it has. Now, as well as affecting the kinetic energy of the substrate, temperature is also going to be affecting the kinetic energy of the enzyme. So as we increase temperature, we increase the speed at which the substrates are moving, the speed at which the enzymes are moving, and so we're more likely to cause these successful collisions between substrate and enzyme. Now, one term I should have brought up previously is enzyme-substrate complex. Now, if we have a successful collision between substrate and enzyme, then what we say is we form an enzyme-substrate complex. If you have an unsuccessful collision, so if this is in the wrong orientation, or if it's moving too quickly, too slowly, or if the substrate isn't complementary to the active site, you're going to have an unsuccessful collision and you are not going to cause an enzyme-substrate complex to form. Okay, so where does this graph come into this then? So what we can see here is we've got temperature along the x-axis, enzyme activity on the y, and you can see quite clearly as we increase temperature, we increase enzyme activity. We increase essentially the formation of the product. And this is because we're increasing the kinetic energy of the enzyme and of the substrate. We're more likely to have successful collisions, more likely to form enzyme substrate complexes. But you can see that we have an optimum. This temperature here is the temperature at which our successful collisions are most likely to occur, and we're most likely to form enzyme substrate complexes. This is because, well, firstly, we have sufficient kinetic energy in both the enzyme and the substrate for them to form these successful collisions. But importantly, we haven't increased the temperature so much that we're actually damaging the enzyme. Now, you'll be familiar with the term denaturing, I'm sure, from GCSE. Now, what this actually means then is that as we increase the kinetic energy of the enzyme, to start with, we have this nice pattern here where you can see that we're increasing the chance of successful collisions. But if you increase the kinetic energy too far, then the enzyme starts to rattle around. And as it rattles, those internal intramolecular bonds are going to start breaking. And in particular, the hydrogen bonds. They're very, very weak. Any excess kinetic energy or kinetic energy past a certain point is going to cause those bonds to break. And once those bonds are broken, the tertiary structure of the enzyme changes, the active site changes, substrate is no longer complementary. So this point here is the point at which the enzyme is denaturing. Right, next up is pH. Now pH, the graph has a similar shape to that of temperature. You can see that we have an optimum pH. That's very important. What does the optimum pH mean? Well, we'll see in a later video that pH is entirely dictated by the concentration of hydrogen ions in a solution. Hydrogen ions are charged. Now, if these charged ions are surrounding an enzyme, then they're going to be interacting with the charged R groups within the polypeptide. Now, if they're interacting with those charged R groups, that means that ionic bonds and hydrogen bonds are less likely to form in the correct places. Some ionic bonds are going to break and some new ones are going to form, but in the incorrect position. Now, if we have the perfect concentration of hydrogen ions around the enzyme, that means these ionic bonds and hydrogen bonds are all going to form in the correct place. So for this enzyme, the pH of about 7.5 gives the correct concentration of hydrogen ions. As you increase the concentration, so you decrease your pH, or you increase your concentration of hydrogen ions and decrease your concentration of hydrogen ions and increase your pH, that is going to cause those ionic and hydrogen bonds to form and break in the wrong places. So what does this mean? It means the tertiary structure of the enzyme changes. It means the shape of the active site changes. It means the substrate is no longer complementary. Different enzymes, though, will have different optimum pHs. Be aware of that. Next up is enzyme concentration. This is pretty straightforward. What we're saying here is if we have a test tube containing our enzyme and containing our substrate, and it's a set concentration of substrate, set temperature, set pH. If we increase the enzyme concentration, the rate of reaction is going to increase. That's pretty straightforward. If you think about it, if the enzyme is controlling the reaction, it's speeding up the reaction. If we increase the concentration of enzyme, we're going to increase the speed or the rate of the reaction. Next up is substrate concentration. So picture this same scenario here. We've got a test tube. We've got a set concentration of enzyme in there. We've got set temperature, set pH. What happens if we increase the substrate concentration? Well, you might think that the graph would come out looking a bit like this. But if you think a little bit harder, what you should realize is that actually, if we've got a set concentration of enzyme in there, we're going to get to a point where we've increased our substrate concentration to such a level that the enzymes are working at their maximum rate. And any further increase in substrate concentration is not going to increase the rate of reaction. Enzymes are very, very fast in how they work, but they are not infinite in their speed. So this point here, saturation point, this is the point at which all of the enzymes within the solution are working at their maximum rate. Any further increase in substrate concentration has no effect on rate of reaction. Right, next up is inhibitors. So enzymes, being proteins with a tertiary structure, have lots of little nooks and crannies into which other molecules other than substrates can bind. Now, the first nook and cranny is actually the active site. So you can see here that we have an active site here. It's a little V shape in this enzyme. And in this case, here is our substrate. And you can see this portion of the substrate is complementary to this shape of the active site here. Now, in this case, we have an inhibitor which also has a shape that is complementary to the active site of the enzyme. Now, thinking back again to random collisions of substrate and active site, if you have a solution that contains both your substrate and your inhibitor, they are both going to be competing for the active site, and through random collisions, one of these two is going to find their way into that active site there. Now, in most cases, a competitive inhibitor is not permanent. So in this case here, you can see the inhibitor has found its way into the active site. Although it has now bound to the active site, it is not in there permanently. So these two molecules are still competing for the active site. And normally it's the case that if the substrate collides with the inhibitor, or indeed if any other molecule collides with the inhibitor, it can be knocked out of that active site and the other molecule could find its way in. So essentially what we're saying is if you need to be thinking about ratios of these two molecules in the solution. If the ratio of substrate to inhibitor is in favor of the substrate, then the substrate is more likely to find its way into the active site and vice versa. The second type of inhibitor is a non-competitive inhibitor. So we've got the same enzyme down here. There's your active site, complementary to the substrate, but we have this second site down here. Now, any other site on an enzyme that is not the active site, but something can bind to it, that's known as an allosteric site. So in this case, our inhibitor here is a little ball, and you can see that is complementary to that allosteric site down here. Now, a non-competitive inhibitor, when it binds to this allosteric site, it causes a change in shape of the active site. So here now you can see that our substrate is no longer complementary to the active site. How does that work? Well, when the inhibitor binds to the allosteric site, it's going to cause changes in the tertiary structure of this enzyme. It's going to do that by causing some of those intramolecular bonds to break and form. So some hydrogen bonds could form, some ionic bonds could form, or indeed they could break. So we've got a change in our tertiary structure, change in the shape of the active site, and now our substrate is no longer complementary. Now, importantly, since these two are not competing for the active site, i.e. they are non-competitive, that means that our random collision theory now no longer works with non-competitive inhibitors. So simply by increasing the concentration of substrate does not necessarily mean that you're going to increase your rate of reaction, because the non-competitive inhibitor will remain bound regardless of the concentration of the substrate. Let's take a look at that on a graph. So here you can see, you've seen this graph before with substrate concentration and rate of reaction. Here you can see normal enzyme, no inhibitor present. And we said that as you initially increase substrate concentration, you increase rate of reaction, but eventually it's going to plateau because the enzymes are working at their maximum rate. So no further increase in substrate concentration increases rate of reaction. Here we've got a competitive inhibitor. Now picture having a set concentration of competitive inhibitor in the reaction tube, and you can see that the rate of reaction with that competitive inhibitor is lower than the rate of reaction in the tube without any inhibitor. But as we increase substrate concentration, the rate of reaction actually finally reaches the same rate as in the tube without any inhibitor. And that's simply down to that competitive theory. The substrate and the competitive inhibitor are both competing for the active site. If you increase the concentration of substrate, it's more likely to successfully collide with the active site compared to the inhibitor, and so finally the rate of reaction reaches that as if there were no inhibitor at all. With the non-competitive inhibitor, though, you can see that regardless of the concentration of substrate, the rate of reaction is always lower than in the tube without any inhibitor. Not only that, but we still plateau as well. And that's because what we're saying here is that all of the enzymes will eventually have a non-competitive inhibitor bound to them, or a set amount of the enzyme will have the non-competitive inhibitor bound and a set amount will not. And so effectively what we've done in this case, you can see the rate of reaction is about half that of the tube without the inhibitor. So we're saying that we've probably inhibited about half of the enzymes. They're now no longer working, and so we have only half the rate of reaction. And regardless of the substrate concentration, we can't increase that any further because the substrate is not competing with the inhibitor because the inhibitor is bound to that allosteric site. Okay, here are your key terms for this topic. So pause now if you want to make a note of those. Loads more free resources on my website, pxsbiology.com. And if you found this video useful, then please remember to like, subscribe, and share.